In this next set of lectures, we want to address mechanistic questions and the answers that we have to date for how synaptic vesicles fuse to release neurotransmitter and how they then recycle, and what we think about that process since the days of Heuser and Rees. But there was a growing body of evidence in support of the vesicular hypothesis of transmitter release, but there remained some doubts regarding whether the recycling was exactly as Heuser and Rees outlined it. Chief among these was the presence of little sub-minis, teensy, very, very small but measurable spontaneous events with less than a quantal size. And this was the work of a gentleman by the name of Kriebel. And the quantal hypothesis was so strong mathematically that everybody just ignored Kriebel's work. And the absence of an endosomal compartment in comparable experiments to Heuser-Rees done by Ciccarelli, so other conflicting EMs. But given that vesicles are seen at the EM level, opposed to the active zones, and that stimulated release was not always reliable, the vesicular hypothesis was thought to be composed of three different separate processes: docking, priming, and then finally fusion and release. Now, docking and priming are hard concepts. Having grown up in an analog world, I think of them in terms of the old-fashioned analog pinball machine, where you put in your quarter and you get your three balls. They immediately come into view. Well, that's docking. Your balls are in place, ready to be played. You pull back the trigger, this very analog cocking of the spring, and you're now primed, ready to have the ball move through the ping pong or the pinball machine maze. But not until you release that lever does the ball actually get into the machine and start bouncing around and doing its thing as it diffuses, or with your help and some tilt, collects points on its way through the matrix of the pinball machine. So those same kinds of thinking were in people's minds as a model for how the synaptic vesicles could align and get ready to fuse with the plasma membrane. Now, the central problem here, though, is that this synaptic vesicle bilayer and the plasma membrane bilayer are both exceptionally stable. Neither one of them both of them are actually in very, very good energetic states. So to get them to fuse and to reform that lipid bilayer, to actually break two bilayers and get them to connect, is energetically unfavorable, and what the processes are in between is quite difficult to imagine. But let's think through that problem. And in the next 4 text slides, what I want you to do actually is to think about this before I tell you what the field is thinking about it. Because once you hear my responses to these questions, you will have lost the opportunity to have done some independent, creative thinking on your own. So I want you to listen to me and then stop playing the video and write down things that you think might be physical or temporal or energetic constraints on synaptic vesicle fusion. So stop the video here and write out your list. All right, so now that you're back, you've done your own thinking, here's what the field has thought: that the mechanisms of synaptic vesicle fusion must have components in them that make membrane curvature change from convex to concave. All right. Then there must be sufficient energy generated in somewhere, either through hydrolysis of ATP or gradients, some source of energy, conformational change, to overcome that very stable bilayer configuration and overcome the energy of activation needed to get those two bilayers to fuse. And if it is energy that's coming from internal sources, what's going to keep that from happening spontaneously all the time? And certainly a small degree of spontaneous neurotransmitter release can be tolerated in the system, but if all the vesicles fused rapidly all the time, then there would be no triggered mechanism for sending that message across the synapse. So clearly there's got to be some kind of regulation. But when vesicle fusion does occur and transmitter is released, it has to be rapid, and it has to be also reversible in terms of being able to reprime the system. And we certainly know that it has to have a calcium dependence because we've seen that evidence already. Now, fusion has got to also occur only between vesicles and the plasma membrane, but not with other intracellular membranes. We don't want synaptic vesicles fusing with the ER. We don't want them fusing with lysosomes. We only want them really targeting to the plasma membrane. So there have to be other proteins or mechanisms for the vesicles to actually do this docking and priming and maneuvering to get into the right position. So a lot of cellular constraints. All right, it's another question to match your thinking to that of the field. Given that set of constraints, what experimental criteria must be satisfied to establish that a mechanism, any mechanism that explains fusion? All right, so you're a reviewer of a paper and somebody writes a paper saying that they've got the answer to how all of these molecules work together to cause vesicular fusion. What kind of criteria do you expect? What kind of experimental evidence do you expect them to present that would be convincing enough to agree that they had indeed found this kind of a mechanism? All right, so pause this and come up with your own list of criteria. Sorry. All right, now you're back. Clearly, we have to identify the protein components, and we have to find those components in the appropriate presynaptic place, not necessarily all over the cell or postsynaptically. And we want to show that these protein interactions or protein-lipid interactions also apply to those same structures. We want to see if perturbations, mutations, or knockout of the proposed components of vesicular release actually prevent the binding and fusion and transmitter release, or in some way maybe alter the kinetics or prevalence or ease of that process occurring. We want to manipulate in the other direction and overexpress the components of vesicular release and see if that facilitates or improves or increases the kinetics of the process. Although one has to be careful here of possible bell-shaped dose-response curves. We want to, of course, make sure that we see calcium dependence to whatever the mechanism is. And we have to be able to understand the energetics, that we can overcome that activation energy to cause the membranes to fuse. And understand that that happens at a reasonable speed. Transmitter release is very, very rapid, but the Heuser-Reis recycling seemed awful slow, so we have to be very cognizant of these kinetics. And then once a pore is formed, if indeed Heuser and Reis is correct, then we have to see the expansion and dilation of that vesicle, and we have to have a mechanism in place for that relaxation and full fusion of the lipid from the synaptic vesicle into the plasma membrane. And there must be some kind of regulatory mechanism in place to keep this from happening except when the action potential arrives at the nerve terminal. And actually, the most convincing experimental criteria one could ever present would be that of reconstitution. That somehow or another you make an artificial system with all of the appropriate components in it, and it acts in a perfect way to mimic what happens inside the cell. Now, reconstitution as a criteria is very, very hard to do, but it is always there as the ultimate in experimental criteria. All right, so before we go on with more text, let's just for a moment imagine what kind of models we might think about in terms of these vesicle membranes fusing to the plasma membrane. So here in all the diagrams, the vesicle membrane is the curved one, and the plasma membrane is not curved. So the idea of there being a proteinaceous pore comes from our idea of channels, which open in response to voltage or ligand. And there's one kind of channel I haven't mentioned, hopefully Eric has. Those are called connexins or gap junctions, where protein half channels, hemichannels, in one plasma membrane align with half channels in the other, an adjoining plasma membrane, and when they perfectly lock in place, then they can be opened to form a passageway that connects the two, what would have been cytoplasmic here, which we have intravesicular and extracellular compartments. So based on this model, connexin-like model, one can think about a fusion pore as being proteins from the synaptic vesicle that match or align to proteins in the plasma membrane to cause some temporarily open fusion pore, which then collapses, bringing the two distinct sets of membranes together into the omega shape that we've seen the EMs for. The other way to think about this pore, though, is not as proteinaceous, but as purely lipid. Now, lipid bilayers are very stable, but every once in a while, some of the lipids can flip. They can move from one side to the other. They can reposition. I mean, no molecule is kinetically ever perfectly without motion. So they could spontaneously, or under some favorable conditions, high calcium being one of them, rearrange the lipids so one could form what's called a lipid stalk. Again, another bilayer, relatively stable. The unstable or weak points are where the curves bend quite a bit, so high radiuses of curvature are not intrinsically stable for lipid bilayers, and the stalk would be at risk to being broken as those highly strained lipid bilayers would then be under energetic pressure to relax into a less curved kind of situation. So one can think of there being a lipid pore as well. And we do know that lipids change phase, that is, they change their relatively stable conformation in the presence of high calcium. So this is not an unheard of kind of a mechanism. All right, just more to think about. All right, so now let's outline and brainstorm some experimental approaches for studying vesicle fusion. So again, stop listening and write out some ideas of your own. All right, glad you joined me again. We've seen some of these. Certainly we want to identify the proteins, and we can take a brute force approach there by isolating proteins from synaptic vesicles and examining their consequences, either in overexpression or knockout, looking for their calcium sensitivity and genetically altering them, expressing them and mutating them to look for important domains, comparing them to databases, etc. Or, alternatively, we could find an analogous simple system. The exocytosis of neurotransmitter and the endocytosis of synaptic vesicles afterwards are no different than any other constituent of exo and endocytosis in a cell. So there has to be other places in the cell where other machinery very comparable exists. And certainly one could look to very simple cells like yeasts, which have very large secretory vesicles. There might be other animal models with very good simple systems to play with. But there's regulated release of hormones by glandular tissue. One of the favorites is the release of norepinephrine by the adrenomedullary cells, or histamine secretion from very large vesicles of the mast cells, which are an immune response cell for inflammation, of a mutant mouse called the beige mouse. So we're talking about beige mice mast cells. Not easy to say. And we certainly have seen lots of experiments where the fusion pore has been mimicked in reconstitution experiments where many of the candidate proteins have been incorporated to liposomes, or little vesicles that we make experimentally with known lipid composition, or into cells themselves by expressing them in a reversed inside-out membrane orientation so that whole cells could be made to fuse, as opposed to little bitty liposomes, to see if we can get a mechanism for fusion to work. All right, last time to write a list and think. What kind of endpoints should these experiments monitor? What kind of techniques, more specifically, should be used? All right, pause a minute and make your list. Glad you're back. All right, so we got all of the tools of protein expression, Western analysis. We have all of the electrophysiological recordings that we can make, looking in both membrane potential recordings and voltage clamp recordings at excitatory postsynaptic currents or inhibitory postsynaptic currents, and applying the quantal analysis, looking for those measures of presynaptic release using the postsynaptic membrane as the bioassay. We can use capacitance measurements, again using the same kind of electrodes with different kinds of electronics. We can use fluorescent labeling of vesicles in order to watch vesicle turnover. We can look at photobleaching and recovery, or fluorescence resonance energy transfer, where and I'll give you an example of that later, for dye unquenching. We can actually look for whole animal behaviors, or muscle contraction, or electroretinograms to show evidence of synaptic function. We can look morphologically at the EM level to see if we've depleted the body, the pool of synaptic vesicles. And we can do ex vivo protein-protein interaction kinds of biochemical assays, immunoprecipitation, yeast-2 hybrid systems, ways to actually try to figure out which parts of which proteins are binding together to make this system work.